For Victor, the first hypothesis is a chromosomal disorder. An array CGH test was performed and was normal. In the meantime, the geneticist learned that the chromosomal rearrangement of the cousin was de novo. Given the mother's wish for a new pregnancy and the absence of clinical suspicion, exome sequencing could be proposed as the next step. The geneticist explains that the absence of the father's sample could be limiting because trio exome sequencing will not be possible in order to filter the genetic changes, variants, found. Anna's geneticist knows that juvenile polyposis is part of hamartoma polyposis and may be related to two different genes, SMAD4 and BPMR1A. Pathogenic variant in SMAD4 is also known to be involved in another rare genetic disease called Rondeau-Osler disease, which is characterized by arteriovenous malformations. One of the main signs in this context is epistaxis. On the contrary, BPMRA1 anomalies do not give this type of manifestation. The targeted analysis of these two genes is thus proposed after an informed consent is given by Anna's parents. For Peter, the suspected diagnosis is cerebellar ataxia. Cerebellar ataxias encompass many disorders with high genetic heterogeneity and numerous pathogenic mechanisms. Some have to be sought with dedicated analysis. For instance, nucleotide expansion-based spinocerebellar ataxias are disorders transmitted in an autosomal dominant fashion with progressive anticipation along the generation. Anticipation in genetics means that the unstable repeated nucleotides tend to expand along the generations, with earlier and more severe symptoms in latter generations. For Peter, the mode of inheritance is unclear. This analysis is thus performed and comes back negative. As more than 100 genes are known to be associated with cerebellar ataxias with poor clinical specificity, a pangenomic genetic procedure called exome sequencing is prescribed.